So welcome everybody to our Agile BioFoundry webinar today. I'm Nathan Hilson. I'm the lead principal investigator of the Agile BioFoundry. I'd first just like to thank Stacy Young within DOE for helping get this Zoom meeting and the webinar set up. I'd also like to thank on the Agile BioFoundry side, Emily Nelson, for helping us get the agenda coordinated and make sure we all know what we're doing. I'm going to be giving the agenda in the next slide so you know what's going to be happening over the next hour, and then I will transition into providing an overview of the Agile BioFoundry. I'd also like to thank DOE, the Energy Efficiency Renewable Energy Office, Bioenergy Technologies Office, and on with us. We're really lucky to have Gail Bentley, who's our technology manager. So thanks to Gail for supporting us for all of the work we've done in the past and the work that we're going to be doing going forward. So in terms of the agenda for this hour, I'll be spending about 10 minutes giving you an overview of the Agile BioFoundry, and then we'll transition into three 15-minute talks, one on the biosensors from Terica, one around machine learning from Hector Garcia Martin, and then one on deep learning from Phil. At the end, we will have kind of like an open mic Q&A for all. We have enabled the chat functionality for everyone, so everyone should be able to chat. Since we don't have that much time for Q&A, I would probably suggest doing most of the Q&A just in chat, and that will give you the opportunity to get as much feedback on your questions as you can. So I'm going to transition now into the overview of the Agile BioFoundry. And the goal of the Agile BioFoundry is to enable biorefineries to achieve 50% reductions in time to bioprocess scale-up compared to the current average of around 10 years. Our objectives and outcomes are the development and deployment of technologies that enable commercially relevant biomanufacturing of a wide range of bioproducts by both new and established industrial hosts. And we'll hear more about some of the hosts in a bit. As a statement on relevance, we are a $20 million US per year public infrastructure investment. I'll give you some more descriptions about what we mean by that in a few slides too. And we'll be increasing US industrial competitiveness, enabling opportunities for private sector growth and jobs. The Agile BioFoundry directly supports the Department of Energy, Energy Efficiency, Renewable Energy's mission and objectives in three kind of high-level ways. So the first one would be decarbonizing energy-intensive industries, not just by making the current industries as they operate now more efficient, but kind of going back and completely changing the way that things are done by starting out, for example, from sustainable feedstocks as opposed to fossil feedstocks. Second kind of category listed here would be around sustainable aviation fuels and generally decarbonizing transportation. And the third area would be diversity in science, technology, engineering, and mathematics. And it was something that we're really excited to be about to launch is a $1 million support for the Minority Supporting Research and Development Consortium in collaborations with us. So we're really looking forward to that. In terms of public infrastructure investment, what I'm showing here on the screen is going to look pretty familiar to you in terms of just a generic subway map. We've also, adapted this from the Bay Area Regional Transit, the BART system around the San Francisco Bay Area. So in dark lines, you might have subways, kind of in the circles, you might have subway stops. But the destinations where people really want to get to, restaurants, businesses, schools, those would be kind of the little dots that wouldn't be directly serviced by the subway, for example, but would be within a short walk or bike ride or taxi ride from where one of those stops would be. So the public infrastructure here would be the subway lines that get people fairly close to where they want to go. But then it's really the private sector, like taxis, for example, that would be getting people to the last mile. Now, through analogy, that's kind of how we're thinking about things too, except we're talking more about chemical or biochemical type of a space. So here, the Agile BioFoundry would be these dark types of subway lines that enables companies to not have to reinvent the wheel, but get pretty close in chemical space to where they want to go. And our subway stops here we're calling beachheads. So these would be intermediate molecules that really wouldn't be the final destination, but pretty close and provide quick access to a variety of downstream target molecules that the private sector might want to be going after. And some examples, in terms of subway lines, we would be having different host organisms. Maybe different host organisms have different metabolic capacities, different bioprocess compatibilities, ability to use different feedstocks. And in terms of, these specific examples of beachheads. Protocatechuate would be one example beachhead, and muconic acid might be one example, or what we call an exemplar, of a representative type of a target molecule. Now, we don't just have this abstract map based on the BART system. We've adapted a metabolic map that was published by Sang-Yep Lee in the past few years to our purposes. And shown here in green would be, for example, our beachhead molecules, in blue, some of our exemplar molecules. And some of these beachheads are established, and some of them are kind of prospecting, as if you would be planning out a new subway stop sometime in the future. In terms of the way that we operate, we collaborate with industry or potentially, other groups, including academics. They would come in with some idea of where they want to go in chemical space. We can kind of support those efforts in terms of technoeconomic and life cycle assessments. I'll show a slide on that in a bit. Host onboarding and development. So you'll see a slide of the host that we're working with currently, but if the industry partner has a new host that they want us to onboard, we have the capabilities to do that. We have our core engineering cycle around design, build, test, and learn. And importantly, in our test space, we do have capabilities around process scale up. And then just a quick slide on technoeconomic analyses and life cycle assessments. So these would really be, helping ourselves or our collaborators understand the economics of the process, as well as, really importantly, for our mission and Department of Energy's mission, looking at those greenhouse gas emissions and the decarbonization types of metrics. In terms of our hosts, we've established within the Agile BioFoundry a tier system for kind of increasing capabilities with each different host organism. We have a manuscript about to submit to make this public, so more people can understand what we mean by these tiers and use it for their own purposes if they find useful. Currently, we have 11 different hosts between bacteria and fungal organisms at tier level 1, which basically means that we can operate with them in a BioFoundry context. And we have 5 hosts between bacteria and fungal organisms elevated to tier 2, which is basically unlocking additional, more sophisticated types of capabilities. In terms of our design, build, test, learn infrastructure, we do go around the full design, build, test, learn cycle. You'll be hearing in the next talk from Terica around some biosensors. And then Hector Garcia Martin will be telling you some more about predictable biomanufacturing and machine learning. And Phil Lively will be telling you about deep learning. If you want to hear more about our capabilities within the Agile BioFoundry, I would refer you to our website, in particular the capabilities page. So in addition to design, build, test, and learn, we have capabilities listed there around scale up and host onboarding and development. So please check out that page. If you're interested in learning how to work together with us, so what are the mechanisms that we could partner with your company, or if you're an academic, we have information there too for you. So please check that out. And to just emphasize, we are a distributed biofoundry across the US. 7 different national labs are participating. And again, we are supported by the EERE office, the Bioenergy Technologies office. And with that, I will stop sharing my screen. And I think we're just at time. So Terica, if you'd like to share your slides and take it away.
Can you hear me?
Yes.
Great. Yeah, hi everyone. Thanks for joining us today. So today I will be talking about increasing the throughput of our design, build, test, learn cycle through biosensors and the approaches that we use associated with those. How to advance? Okay. So the design, build, test, learn cycle actually has the potential to be very high throughput. There are a number of advances that have been made all throughout this cycle that allow us to both move through the cycle relatively quickly in some cases, and also parallelize the cycle using combinatorial and library-based approaches. So in the design space, there are ways using combinatorial and computational design to make very large libraries or very large sets of variations in strains or vectors, for example. And within the build space, there are, again, opportunities to make, large libraries up to 1 million different variants or more in organisms, at least with high transformation efficiency. And in Learn, if you're looking at things one by one, Learn can be very slow. But as I think you'll hear from Hector next or later, and there are other, and Phil, there are various ways, obviously, to computationally increase our Learn throughput. And in test, it can be really varied. So if you're just looking at single flasks or doing your biochemical or analytical assays in sort of a traditional way, it actually can be pretty slow, down to, if you're thinking about, looking at 96 well or even 384 well plates, it can be, looking at down to the thousands. And so we are really interested in enabling all of the higher throughput of this cycle, but specifically trying to improve the throughput of test. And so focusing on library-based screening, where we can look at, at least libraries of 10,000 to up to 1 million variants in one single tube. And there are a number of ABF capabilities that enable this higher throughput. These come from a variety of the national labs associated with the ABF. In the design space, we have expertise in computational protein and library design. In the build space, there are automated build tools that are being worked on, as well as the development of approaches for building larger libraries, especially in non-model microbes where the transformation efficiencies may not be as high as your traditional favorite laboratory microbes, such as E. coli. In the test-based space, which is where I'll talk a lot about today, we have a lot of expertise in developing fluorescence-based assays and developing fluorescent proteins for these types of assays, custom biosensor development, which Nathan mentioned briefly, and then using things like flow cytometry and microfluidics for that rapid screening, and then also even other novel approaches for screening libraries, not just for survival, but directly looking at the productivity of your strain. And then on the Learn side, which again, you'll hear more about today, capabilities and tools for identifying enriched sequences in populations, and all in addition to regular clonal sequencing that then ideally can be fed into predictive models. So the general scheme of the type of approach I'll be talking about today is shown on this slide. And there are a lot of steps, but it's a cartoon, so hopefully it'll be pretty easy to follow along. The idea is that we have a library or a population of a lot of different variants, again, in one test tube. And these variations can be just a single protein. It can be the genome scale. They can be on plasmids, or they can be integrated within the genome. But the idea is that you have a lot of different genetic variations in one spot, in one test tube, and you want to try to figure out how to parse those into what is interesting and what may be less interesting. And the way we do that is we tie phenotype to fluorescence. So most often it's some type of fluorescent reporter, and whether that's just straight up GFP or the GFP is associated with a biosensor, for example, it depends a little bit on how we set up the experiment. But the idea is then you have your library, if you were to look at it, has differing fluorescence intensities for the different phenotypes. And you can see that then if you put this type of library on something like a flow cytometer. And so if you have a negative control, that's going to have very low fluorescence, and maybe you have a positive control that has very high fluorescence. And then your library ideally is going to have this kind of broad smear of different phenotypes. And in this graphic, we actually intend to fix. The idea is that your smear would go even past your best positive, so that ideally you're pulling out even more improved variants than what you've started with. But nonetheless, the idea is then you have lots of different phenotypes, and then using something like fluorescence activated cell sorting, you can pull out variations on subpopulations that have these different phenotypes, sequence those subpopulations as pools, and then look for enrichments in the different sequences to tie that phenotype to changes in the genetic content of your cells, and then feed that into machine learning. And so that's the kind of general approach that we are taking for a variety of different efforts. And so as an example, we've applied this for developing a synthetic biology tool that we call cis repressors These are RNA-based regulators that help regulate messenger RNA translation. They're sort of an extension of the ribosomal binding site, and what they do is they form a hairpin that to varying levels occludes the ribosomal binding site and then either allows the ribosome to get in or disallows the ribosome to get into varying levels such that you get different levels of protein expression. And so in this particular example, these hairpins are upstream of superfolder GFP and on a vector. And what we did in this particular work was we took a library where we had different variations within the hairpin that we expected to allow it to open or close to different equilibria in the cell. And we were able to show that, in fact, that library did have this very broad distribution of fluorescence intensities. And then we used fluorescence activated cell sorting to pull out 8 different subpopulations. When we regrow those, in fact, they do have different fluorescence intensities. And when we sequence those subpopulations, we were able to identify sequences that were well represented within those sort of subpopulations and not so represented in the others. And when we pulled those out and made those individual variants, we were able to show that we can tune the fluorescence intensity of GFP, really well using these different sequences. And we were able to show that not only does this work for GFP, but it also works from chloramphenicol resistance. We showed that this works on plasmids as well as in the genome. We've demonstrated it across a couple of different, I think 5 different organisms now, 2 of which are in this paper. And we also tied it to mucinate production in this particular example and showed that, using these riboregulators, we can tune the cell growth as well as mucinate productivity. And so the outcome of this was that we were able to use this approach of creating a library, tying it to fluorescence, and then using cell sorting and pulling out different variants to identify a whole suite of this type of tool without having to know a priori which sequence was going to give us the best result for these intermediate phenotypes. And this allows us now to have this toolbox that allows us to tune translation across a number of different systems. And similarly, we use the same type of approach when we are developing biosensors. So we are focused on using transcription factor-based biosensors. Transcription factors occur in native cells, and what they do is they bind small molecules in the cell and either repress or turn on gene activity. And so what we do is we set up we harness that, piggyback off of that, and set up an artificial system where our transcription factors turn on GFP in the presence of a small molecule. And so if our small molecule is a bioproduct of interest, such as mucinate, then we can identify cells that have either varied or very increased concentrations of mucinate, because those cells will make more GFP and therefore they will glow more green. And so in order to do this, we have to make the biosensor first, and we use the same type of approach, where now we're making, instead of variations in an RNA, we're making actual variations in the transcription factor and the DNA to which it binds. And those libraries are then screened for fluorescence output, and then we can pull out the brightest culture, subculture. And then we can identify the sequence of that biosensor, and we've been able to change the specificity of biosensors. We can tune the sensitivity of the biosensor to different dynamic ranges, depending on where a person is at in their strain development for a given product. So if you're making very little product, maybe you want a very sensitive biosensor, but if you're pretty far along in your metabolic engineering, then you may need a less sensitive biosensor so that it's not saturated by the time you get to the studying of it. We've been able to tune these to different hosts. And so we've developed essentially a general approach for moving these sensors across organisms and for detecting new molecules. And there's a couple of papers. This is just actually a subset of papers where we've described some of this work. And then similarly, we can actually apply these biosensors to look at metabolic pathways and production of bioproducts of interest. And so we have a couple of different examples where we have, instead of messing with the transcription factor, now we have the biosensor of interest, and now maybe we want to manipulate the enzyme or on target or off target enzymes that might affect the production of our small molecules, such as mucinate. And so then again, we're making, using computational protein engineering to make gene libraries, and then pairing that with the biosensor system in order to screen which cultures or which cells make the most product of interest. And we've been able to show that, again, using this system, we're able to make higher cells with higher productivity and reach those max productivities in less time. And we've also demonstrated that we can use this to improve enzyme productivities in a certain pathway by reducing product inhibition. We've identified new off and on-target genes tied to improved mucinate titers, and also use this to start looking at transporter sequences for different uptake molecules that might be important for bioprocesses. So what else could we do with this? So where are we going next? Again, this is just the same graphic as a reminder that we're doing these library approaches, and then we're trying to get to the sequencing and machine learning piece. And the examples that I just gave you did not have a heavy machine learning piece. It was all very either pick single colonies, or in the case of those riboregulators, we really looked at those sequences by eye. And so what we want to do next is look at different types of libraries. And so we're really interested right now in whole genome library screening. We have some experience in the past with doing adaptive laboratory evolution to change the diversity of our populations and do screening. That was published in the Bentley paper. And we also have some additional adaptive laboratory evolution experiments that we've done that we're kind of wrapping up currently. But in the meantime, we're developing the same type of process, trying to demonstrate this entire process for other types of knockdown and knockout libraries. So we have 3 different types of libraries we're working on sort of in parallel across the ABF. One is a trans repressor base, which is roughly based on the cis repressor RNA tool that I just described. We have CRISPR-I knockdown libraries that in the ABF, and then we're working on these RBT and seq knockout libraries as well. And of course, all these types of libraries have been used and are being used pretty regularly, but mostly associated with fitness or tying your product of interest directly to fitness. And so what coupling this to sensors does is allow you to directly detect your molecule of interest instead of having to tie it to fitness and the other complications that may come with that type of more indirect screen. And then for integrating with Learn, this is something that we're really excited about, that we're focused on trying to get really off the ground in the near future. So instead of just sorting for the brightest green, the best producers, now we're really interested in pulling multiple populations the way we did with that RNA, the cis repressor RNA experiment. So we want to screen for productivity, variations in productivity, essentially, and pull out subpopulations of sequence variants that are tied to something like, say, mucinate productivity, and then do the sequencing and then feed that information into our into the Learn team. And the reason for this is that for machine learning models, it's important to know not just what works, but also what does not work in order to better bound those models. And so library-based screening is a really great way to support this, and again, allows us to significantly parallelize the DBTL effort that we have in the Foundry. And that's the end. Thanks to, there's obviously a lot of people involved in this, so thanks to that BioFoundry team and to everyone who is listening today.
Great, thank you, Tarika. Hector, if you want to start sharing your slides, and everyone, if you have questions for Tarika, feel free to put them into the Zoom chat, and Tarika can be watching for those while Hector presents. Go ahead, Hector.
Okay, can you see my slides now?
Yes.
Okay, great. Okay, well, thank you for the opportunity. I'm here to talk about machine learning and predictive manufacturing in the Agile BioFoundry. The first thing I'd like to say is that really machine learning has applications to the full synthetic biology spectrum, all the way from design which product to satisfy a societal need, how to do the DNA design of the pathways, how to engineer the biological system and optimize it, all the way to high performance system, to how to scale it up and how to downstream processing to get the final bioproduct. And we discussed some of these or many of these applications in our review from metabolic engineering, and you can have a look at that if you are interested in this topic, or you want to learn, you want an introduction of machine learning for metabolic engineering. I'm going to concentrate right now on this part over here in optimization the system or doing DNA design. And the reason that we focus on machine learning is because this transition from learn to design is the main bottleneck for the DBTL cycle, the design, build, test, learn cycle. We have for design, we have a variety of tools like deve, Reven, or the Cellegen design module. We're starting for making strains. We're starting to have a lot of CRISPR tools to modify strains, and the ability to synthesize DNA is really going down in an exponential way. In test, we're starting to see things like the high throughput flows for standard mass spectrometry, but also the kind of fluorescence biosensors that Taraga was talking about in the previous talk. But in learn, often what we have is just a lot of data, and we have to think. This involves sometimes things like kinetic models or scale models of a type, but they often don't work as expected, and how to reconcile the model with the data involves thinking. And that's perhaps great if you're in an academic environment where you have like 5-6 months and a postdoc to think, but often in the industrial environment you don't have that time. And the good thing, or the good news, is the machine learning really provides algorithms that systematically improve with more data. So it doesn't involve someone having a happy idea. The algorithms that we have, they are designed to improve systematically with more data. As you create more and more data, and it tells you where to look for to get that data to make them better. And I'll give you an example of that. So that's the advantage of machine learning, that it really is able to couple this test to design in a really systematic way that doesn't involve months, but can be done in days. Now, ABF, to really support these abilities like machine learning, has created a whole set of computational infrastructure for predictive manufacturing, including like the inventory of composable elements to keep the information on strains. Also things like the experiment data depot, where you can put all your omics data and all your experimental data and be able to visualize it and then download it into machine learning algorithms that will really recommend the next experiment. And I'll talk about art in this case, and in the next talk, you will see some of deep learning techniques that Phil Label has been applied. In terms of art has been decided to suit the synthetic biology needs. So art can work with very few instances, and I'll show you an example with which got results with as little or as few as only 27 instances. R provides uncertainty quantification, so it tells you whether the prediction that it does is good or not, or whether or let's say whether it trusts that prediction a lot or not. So instead of telling you this is my prediction, it gives you the whole probability distribution of the prediction from which you can quantify the uncertainty. If it's very localized, it's a very certain prediction. If it's very broad, it's a very certain prediction. And also R provides not just predictions, which is what a lot of machine learning methods do, but recommendations for the next step. So it really doesn't just take that information to predict what's the outcome, but uses that predictive power to recommend the next design cycle. And let me give you an example of that that's been published in a couple of papers in Zhang et al. And Radivojevic et al. A couple of years ago, in which we're working for people from the Center for Biosustainability in Denmark to engineer yeast to take glucose and convert it into a tryptophan. And in this case, they had a particularly interesting technique in which they were able to use CRISPR to create libraries of promoters. They were able to choose, let's say, 5 genes here and 6 types of promoters and make whole libraries in a high throughput fashion. And that kind of high throughput capability is something that we can leverage through machine learning. Now, in this case, we used a mixture of machine learning and mechanistic models. This is the reaction set all the way from glucose to the glycolysis, hyperglycolysis, and pentose phosphate pathway, all the way into tryptophan. And we use genome scale models to figure out or to predict what are the 5 reactions that will be most likely to impact the productivity of tryptophan. And then we had a library of 6 possible promoters for each of these 5 genes. And as you can see, if we did all of them, all the 6 promoters For all of the 5 genes, we will have around 8,000 combinations, which is quite a lot of combinations to do and experimentally quite challenging. What we did is we did only a few of them, around 5% of them, and from that we were able to use that data to extrapolate to find what are the promoters that will give you the highest productivity. We did this with the automated recommendation tool that is trained with data. In this case, the training data is going to be an input and a response. The corresponding response, the input is the promoter combination for gene 1, promoter 1, for gene 2, promoter 7, for gene 3, promoter 15, and then the corresponding amount of tryptophan productivity, which is what we're trying to optimize here. We have a variety of instances of that that is used in R to train the model. And in this case, we had 264 combinations out of the 8,000, or 3.4% of all these possible combinations. And even though this was still only 300 combinations, it wasn't 8,000, we still needed a high throughput way of doing the measurement of tryptophan productivity, which was done through a biosensor that created a certain amount of green fluorescent protein depending on the amount of tryptophan. And that is what really enabled the high throughput data set to do this. With this data, we created a predictive model that is probabilistic, as I said, so tells you this promoter combination has a 10% chance of producing 10 milligrams per liter, or a 20% chance of producing 5 milligrams per liter, and so on. And then it uses this predictive model to do recommendations for the next cycle. So if this combination of promoters produce this response, try the 3rd promoter in the gene 1, the 9th promoter in the gene 2, and the 12th promoter in the gene 3 to create, to produce this amount of tryptophan with this probability distribution. Now, in this case, well, we started trying to do 696 well plays, which was supposed to give us around 600 strains, and in triplicate around 1,800 samples. But of course, this growing the strains and constructing them had some problems, because this is biology and there's always things that don't necessarily work. Sometimes we didn't have the genotype information of which genes which promoters were put in each gene. Sometimes there was a failed assembly of the CRISPR method, and we weren't able to put the promoters that we were expecting, even though we were able to read the genotype information. Sometimes there was a growth threshold that did not pass, so we thought we couldn't do any sequencing. Sometimes we weren't able to get rid of the complementation plasmid, and sometimes we didn't get a single population. So all of this attrition of data ended up in producing just a third of the initial expected data, around 792 instances or samples in total, which is 264 combinations of promoters, which is 3% of all the possible ones. In order to use machine learning, you need to use a target. You need to figure out what you're trying to optimize. In this case, we use tryptophan productivity, which is one of the things that is least intuitive to increase. People know, metabolic engineers know, have an intuition on how to increase yields and titers, but productivity is something that is much harder, and that's why we tried that. In order to do that, we measure the change in GFP, that is the measurement in tryptophan, and divided by the time that it took to do that, and that's our productivity. We tried to choose a time that wasn't too early, so we didn't get lag effects, but it wasn't too late, so we didn't get into aeration effects that could be not maybe translatable to other fermentation, other ways of doing fermentation. This is the data, the actual productivity, the change in GFP or tryptophan per unit time. This is for the 1st gene, 2nd gene, 3rd gene, 4th gene, and 5th gene, and each of them have different. We have a choice of 6 different promoters. You can tell there is no clear trend. It's not clear whether you should use the 1st promoter or the 6th promoter in gene 1, because they all span all the productivity sample. The only exception maybe is this gene 3. These promoters here seem to produce the higher productivity. But other than that, there is no way that you can choose just by looking at it, this promoter, that promoter, that promoter, that promoter, and that promoter. So that's why we are using machine learning to do that, because machine learning is particularly good at ingesting all this data and figure out the nonlinear relationships between the different promoters. Now, in this case, we were lucky to work with people who really care a lot about the data quality, and you can see that in the replicates. So this is some of the replicates. So we had 3 replicates for each of them. is replicate 1, 2, and 3. This is the histogram of all the productivity values, and this is the replicate 3 versus 1, replicate 2 versus 1, and so on. If there was perfect replicability, all the results will be on the diagonal. Most of them are, but there are some, particularly this one, that are really problematic, and that's where they were eliminated. So we filtered out around 15 replicates with high errors because that will throw the machine learning approach off. And then Art ingested all this data and suggested new promoters, and suggested 15 of those, and said, well, try the third promoter in gene 1, the 8th promoter in gene 2, and so on. And you should expect this amount of productivity with this product distribution. This is the mean for each of the predictions. And this over here is the highest amount of productivity that obtained so far. So this tail of the product distribution tells you what is the probability of exceeding the productivity that you had so far. So how did it work out? Well, quite well. We were able to increase productivity 100% over wild type over the base strain and 70% over the best in library. These are the observed productivity through the fluorescence proxy, through the GFP, and this is the predicted productivity. This is the initial base strain that we started with. This is all the library that we did with the 272 different promoters that used provided by CRISPR method. And these are the R recommendations. And as you can tell, the best recommendations improve productivity 70% over the best in library and around 105% over the base strain. And this was just one DBTL cycle. Now, as you can tell, this is just an fluorescence proxy. How does this stand when you actually use the HPLC to measure the amount of tryptophan? And we did that, and the base strain, interestingly, had none of the strains in the library actually surpassed the base strain, but the recommendation done by ART actually increased production over the best in library and base strain by 43%. And this was the result of one DBTL cycle. As I said before, we can get all these new recommendations and the data of the measurements, put them back in ART, and in a matter of 20 minutes, you can have new recommendations and keep increasing in a systematic way without really having to break your head. So this is what is called active learning. So to recapitulate, ART is ideally suited for synthetic biology needs. You can work with few instances, and in this case, I didn't talk about the case with only 27 instances. I do it in a minute, but we did show that how we provide uncertainty modification and how we provide recommendations, not just predictions. Instead of working with a few instances, you can see an example in the ART paper that Radivojevich et al., where we were able to take 27 instances from a laminin producing strain, and we were able to make predictions for the second DBTL cycle that were quite accurate. And indeed, you will see that the recommendations we did with ART are very close to the ones that were experimentally tested in that project. So we can really leverage a low amount of instances, as low as 27 so far, and really create meaningful recommendations that can guide the metabolic engineering in an effective way. Well, if you're interested in ART, there is a whole webpage, art.ldl.gov, that has tutorials and a paper that explains how to use ART with synthetic data and how to use it with EDD and ICE, and you're welcome to use it. There is also ART frontend at art.agileBioFoundry.org. If you're not interested in coding, you can just upload your data, and then the server will send you your recommendations that you can use as you please. That's it. I mean, synthetic biology can do a lot of things, from biofuels to biomaterials and bioproducts to a variety of different things. But the problem is that we can't really predict what's going to happen with synthetic biology, so it's really hard to design. Machine learning can help that, but it needs tons of data that can really truly only obtain effectively through automation and the high throughput methods that Taraka had been showing in the previous talk. And that's it. I really want to thank for all this work to the people in my group, but also all the different groups in the national labs, both in Berkeley and other ones that have really made all this work possible and make the national labs, the interdisciplinary environment that is ideal for this kind of work. And if you want more details on these capabilities, you can always go to agileBioFoundry.org/capabilities and see all our design, build, test, and learn capabilities. I only had 15 minutes today to show this, but there are many other things that the Agile BioFoundry can offer for your company. Thank you very much.
All right, thanks, Hector. And if people have questions for Hector, please put those in chat for now. And Phil, if you want to share your screen and get started.
Thanks, Nathan. Also, thanks to Emily and Stacy for help organize this webinar and for this opportunity. Today, I'm expanding on what Terica and Hector have told you, and I'm going into another capability of the ABF, where we're using deep learning for improvements in strain and process designs in biomanufacturing. So just to go back to the engineering cycle that the Agile BioFoundry has adopted, I want to just highlight right now that this is an effort that is highly integrated between the Learn team and the test team to use advanced data sets that are coming from our test capabilities. And you'll see that we're not only expanding these capabilities, but we're integrating stronger with design and with build as we go forward into future iterations and improvements using Learn within the Agile BioFoundry. Everyone has done a pretty good job of letting you know how to find more information about our capabilities. And I'll just reiterate, Learn can be found here on our web pages. And both Hector and I are talking about AI approaches. And you can find out more by going within these subpages. But also today, I wanted to let you know that the deep learning is actually going to take advantage of a lot of other Learn capabilities within the Agile BioFoundry to employ mechanistic models, metabolic models there, regulatory models, and in order to understand the predictive outputs of these models, we have complex data visualization that can help in those regards. So we have adopted over the last few years within our aspect of the Learn team an ecosystem for strain improvement. And this is depicted on the slide where we're going from the left side, where we're organizing builds and strain engineering efforts, all the way through test and using models to understand how information flows as processes scale from a miniaturized variation in the bottom part of the middle part of the graph up to scaled analytics that are found in pilot level facilities, then through production phases and clear dissemination or determination of the phenotype of engineered strains that we are evaluating within our design and host target teams. And so we'll go more into this and how deep learning is applied. What we're actually doing is building relationships between these different data types at different scales and different levels of evaluation of the organism, and then building artificial neural networks between these layers and stacking these layers such that the output of one of the layers becomes the input of another. And so you have relationships between them, and you can very clearly then go between the start of this process to the outputs, and in the end be predictive of what new modifications would do if they're evaluated in the same way. So again, the deep learning exercise has mainly leveraged large omics data sets that are available from our test team at Pacific Northwest National Lab. So here we're talking about transcriptomics, proteomics, metabolomics, both internal metabolites and external metabolites. These are all available and used as layers in deep modeling approaches in combination with information from our build and strain engineering efforts, all the way to then metadata on what the phenotype is doing with advanced analytics on the product production end. So here then, say it's in a different way, show it diagrammatically, we're leveraging relevant biological frameworks to understand what the engineering is doing on an organism level, to understand what's being up and down regulated, what proteins are being produced in larger levels or downregulated within the cell, and how all of these components in the cell interact and work together to change the output of this bioprocess and influence the final titers, productivity rates, and ultimate yields of these processes. and it's iterative. So we'll go through, work with the teams, get a data set, learn from it, predict, and then use these predictions in new rounds of design, build, test, and learn, and use the new data then to reinforce our training data sets to improve our predictions, and then reevaluate the performance to see if we're getting better or need to build in a new type of data set to help with what we're looking at. These approaches are being adopted from other fields of use where it's very helpful to use information theory as a modeling scaffold, where we're taking inputs coming from a fermentation process and using transmitters, channels, and receivers and to decode the information and use our models that are expanding and adapting from a metabolic understanding of organisms, understanding how regulatory networks within the organism turn off and on sets of genes and influence what they're making, how they're growing, what they're utilizing as feedstocks, and then know in the end for our needs how that's affecting them in a biomanufacturing context. And so this approach can be used in a complementary way to what Hector was describing as optimizing a production pathway, but it has really shown to excel in identification of off-pathway engineering targets that would help turn off or modify pathways that are no longer needed of that organism, maybe to survive in nature, as they're now being repurposed in a biomanufacturing context. And this is also important to consider in an integrated bioprocess environment. And this is where these deep learning approaches are really starting to excel. So here's the generic approach that we use for deep learning. We are setting up what you'll hear referred to in this presentation as a learn-friendly experimental approach, where it's usually guided by giving experimental observation or the inability to have a good experimental observation where you know what you want to do, and work with experimentalists then to define an experimental goal. Do you want to increase the titers, the rates, the productivity, or the overall yield of the process? And then design experiments around these goals, ensuring that you have a matrix of experimental parameters that distinguishes between different mechanisms that may be leading to the improvements or negative effects that you're observing, and a range of phenotypes like Terika was talking about. So we know not only where we think we want to go with first principles understanding of that approach, but also where it's not encouraged to go. And those are very important distinctions. From that, then we can identify a modeling approach, design modeling parameters, and most importantly, then interact with the test team to make sure the requirements that we're getting and training data sets are going to be able to feed our modeling approaches. So some lessons learned, and we have learned in some of the first iterations and years of going through this process, is that establishing rapport and a common language is very important to build a really great working relationship with engaged experimentalists, and that goes a long way. Also, productivity and the outcomes, the predictive outcomes, are greatly enhanced through the establishment of learn-friendly experimental designs, like we've talked about here on the left-hand side of this slide. So here's one example of many that we've used these approaches and these learn-friendly experimental guidelines to work on improved forms of biocatalysts within Agile BioFoundry. So here's an example where we're using Pseudomonas putida to produce muconic acid. Terika talked about this a little bit with her biosensors. And the observation that we had in this one case is that a single gene deletion led to differential regulation of more than 18% of the transcriptome and more than 38% of the metabolome. And so we established a goal then to understand what are the regulatory mechanisms which small changes in the gene profile of this organism lead to very large changes in the metabolic structure of the organism. And through several rounds of these deep learning type approaches with predictive outcomes and going through this learned ecosystem in different ways, we have led to the understanding or the identification of more than 30 different improvements that could lead to strain modifications and increased titers, rates, and yields for this organism. Here we have over 30 regulatory genes, some of which are important to the production pathway, others that are influencing growth of this organism or the utilization of feedstocks by this organism. We also are studying transportomes through artificial neural network models, and we have looked at 3 transporters that increase the export of the product from the organism that decreases toxicity. And there are also 32 gene targets, many unannotated, that were proposed to increase growth or product titers. And these predictions are in various states of validation and reinforcement, but it's a real key example of how you can actually layer these modeling efforts with AI approaches and end up with deep learning with predictive capability that can improve your biomanufacturing processes. You can see that our modeling approaches have evolved through the start of some of these projects to be relatively simple, and we can make them as complex as we need to understand and reach the goals that the modeling team and the experimentalists have agreed upon. So here's an example where you're going from a simple information network that mainly is focused on regulatory networks and relates them to both levels of metabolites outside the cell and inside the cell, to one on the right where we're looking at interactions between different types of information within the cell, not only external but internal, how the protein levels are varied, and how that influences then the production of the product that this host has been charged with making. We have active academic industrial projects where these approaches are being utilized, and there's just a sampling of some of the projects or the goals of the projects that we've worked on in this capacity. One is identification and maintenance of prolonged periods of maximum product synthesis. If you have an organism that goes into a stage where it's producing something in a steady state, you want to keep it there. And can we learn how to do that and overcome departures from that steady state? Yield enhancements through elimination of unnecessary off-pathway metabolic processes. This is going to really highlight your yields ultimately, and there are many examples of where these enhancements are needed. Utilization of complex feedstocks that have low, zero, or negative value is an area that is growing. Also, biocatalyst optimization, where you might have a step or two in your pathway, or maybe an off-target pathway you want to eliminate, or in the pathway process you want to optimize. You can do this by studying a range of biocatalysts and how you can optimize that. And lastly, as an example, assembly stabilization and function of a large multi-cylinder unit macromolecular assemblies. You can look at how these assemblies and subunits of them are synthesized independently and come together to form a complex that actually results in high levels of the product that you're interested in manufacturing. So with that, toward an end and future looking, we want to use these deep learning approaches to end up with a learn-guided intelligent biomanufacturing scheme where we can actually expand on the type of models that are included within our deep learning exercises to, on the front end, know more about the feedstocks and whether or not you can get away with feedstocks that are of reduced value or have inherent variability. And toward the other end of the spectrum, on the advanced output side of these value chains, you want to know if you're producing this, can you get it out of the system in a downstream processing scenario that is effective? And do we need to include anything that would enable downstream processing early on in our understanding of the process, or the global process in general. And these are things that we're considering incorporating in newer iterations of this deep learning approach. And lastly, there's the potential to include economic and environmental modeling efforts in the deep learning approaches to know how impacts in earlier stages of these steps in the biomanufacturing may impact them, and you could factor those in early on if it is of value. Lastly, we talked about this learning ecosystem and how it's been functioning for us in the Agile BioFoundry in some example processes and expansion into collaborations with academic partners and industrial partners. However, there's a lot of potential for ecosystem expansion, leveraging things that Terica have reviewed and also Hector has talked about, where we'll be able to learn with larger, more complex and complete data sets. Where here we can use library of strains going into our evaluation phases, maybe libraries of engineered biocatalysts, utilization of information from pan-genome analyses, so we know what an organism is needed for its normal function and survival in a particular environment. But many organisms have large expansions of their genome that allow them to really excel with different product classes and function in varied ecosystems. And so can we utilize them and take advantage of them in the future in our deep learning approaches? As Hector and Terica have alluded to, miniaturization of cultures and bioreactors is going to be really instrumental in moving our processes forward. One example is through microfluidics, where in the upper right, we're showing that you actually could evaluate cells in small little test tube environments that might be as low as 70 picoliters. And in the upper right-hand corner, in those cases that they're little droplets, those little microfuge tubes are holding millions of bioreactors within them. And if these can be leveraged and feed AI evaluations of these strains, then we really have something, and we're getting very close to being able to use them in the Agile BioFoundry. So certainly important to talk about higher throughput means of strain evaluation. As Terica alluded to, biosensors have helped the Agile BioFoundry in many ways, not only with what Terica talked about, Hector talked about them, and we're utilizing them in our deep learning approaches. Flow cytometry is going to be very helpful as well. And especially if you can couple that with high throughput sequence-based analytics that are very available today. And in addition to that, high quality, rapid genome resequencing efforts that would able to capitalize and utilize adaptive laboratory evolutionary insights in deep learning approaches. So we can really understand when you put selective pressure on an organism and evolve into something that's creating something more efficiently and for lower cost, how then we can get this information very rapidly into our AI approaches so we can capitalize that going forward.